Forms of cell transport that require energy are of course referred to as being active. In this case, an active version of transport is simply called active transport. It is exactly the opposite of everything that you learned about diffusion. Whereas diffusion was down, passive, and non-mediated, active transport is each of the choices in opposition. It is up instead of down, it is active instead of passive, and it is mediated instead of non-mediated. If you look at the diagram, you can see an area of high concentration of solute here, an area of low here. And so we're going to have particles from the area of low concentration move toward the areas of high concentration. So they will go from where they are less crowded to where it is already more crowded. It is, of course, an active process. It says so right in the name, which is why you see a representation to ATP here. Your body will have to expend your energy to basically pump these solute particles against the concentration gradient or up the concentration gradient. And finally, it's mediated, which is why you are seeing these large proteins acting as carrier molecules, moving those particles from one side of the cell membrane to another. There are two other forms of cell transport that are active, and they are very similar to one another. One is called endocytosis, the other very similarly called exocytosis, and really the difference between them is directional. Endocytosis moves particles inward, exocytosis moves them out of the cell. And in both cases they are active. They are going to require your body to use energy. And in both cases, they're going to involve the formation of a vesicle. So you see a vesicle here. It's nothing more than a sac within a cell in which some sort of particle is being transported. And you can see how that vesicle forms. Here, you're looking at the cell membrane of the cell. And here you're seeing particles that are outside the cell. The cell membrane starts to dip or fold in. The correct terminology here is that it invaginates. And as it does so, you can see these portions here and here getting closer to one another till eventually over here they touch and that whole sac basically pinches off. In other words, the vesicle, that border on the outside here, is made of what was a minute ago the cell membrane itself. And now it contains within it the particles that a minute ago were outside the cell. If we take that same process but work essentially in reverse, you have exocytosis. The vesicle has already formed inside the cell. Particles that the cell probably produced are inside that vesicle, and you can see that it merges with the cell membrane and essentially becomes part of the cell membrane, and in doing so is able to open up and release the contents of that vesicle out of the cell. It has exported it.